When we picture human anatomy, we usually default to something really structural, like permanent.
Right, like looking at an old medical textbook.
Exactly. The skeleton is drawn like architectural blueprints. The bones are rigid, the heart has its defined chambers, the lungs have specific lobes. We take a lot of comfort in the idea that our organs have a permanent, fixed geometry.
I mean, it definitely makes the body easier to understand, thinking of it as this biological machine made of solid parts. But that model completely falls apart when you look closely at the brain.
Oh, completely.
Because the brain isn't just some static wiring board housed inside your skull. Its very architecture breathes, morphs, and, well, it physically shapeshifts depending on your state of consciousness.
Which brings us to the mission for today's deep dive. Welcome, by the way, to you, our listener. Today, we are exploring a truly mind-bending 2026 paper from the journal Neuron by Dang and colleagues.
Yeah, a brilliant piece of research.
The title is a bit dense. It's called Astrocyte Cell Volume Dynamics Across Cortical States and Transitions.
A mouthful, for sure.
Right. But the core mystery we are trying to solve today is this. What if your brain physically changes its shape when you fall asleep? Okay, let's unpack this. We need to completely discard the idea of brain cells as stiff, unyielding wires.
So no more computer analogies.
No more computer analogies. Instead, I want you to imagine the inside of your head as a dense forest of microscopic sponges. And these sponges are actively inflating and deflating as you go about your day.
That sponge analogy is actually the perfect starting point. Because when we talk about the brain shape-shifting, we aren't talking about the neurons themselves.
Right, the neurons get all the glory.
They do. Historically, neuroscience was so heavily focused on neurons, the cells firing the electrical signals, that astrocytes were mostly ignored.
And an astrocyte is what exactly? For the visual here.
So there's star-shaped glial cells. Glial basically means glue. The assumption used to be that they were just passive structural glue holding the important stuff, the neurons, together.
But this paper kind of blows that apart, right?
Totally. This research proves they're incredibly dynamic actors. But to understand their impact, we really have to look at the interstitial space.
The interstitial space. Okay, break that down for us.
Think of it as the fluid-filled gaps, like microscopic highways that run between all the solid brain cells.
Okay, so the landscape consists of these star-shaped astrocyte sponges and then these fluid-filled highways running between them.
Exactly.
My immediate thought when reading the methodology of this paper was, how is it even possible to watch a single microscopic cell inflating inside a living, breathing animal?
It's incredibly difficult.
I mean, it sounds like trying to measure a single blade of grass growing while you're flying over it in an airplane.
That's a good way to put it. The technical execution here is staggering. The researchers utilized in vivo 2-photon microscopy.
In vivo meaning in a living subject. Mice, in this case.
Right, living mice. Because normal brain tissue scatters visible light, it just looks like an opaque gray mush. They used highly concentrated infrared lasers.
Because infrared goes through the mush.
Precisely. Infrared light penetrates deep into living tissue without causing heat damage, so it allowed them to look directly into the cortex of living mice in real time.
But infrared light alone doesn't single out the astrocytes, does it? You'd still just see a dense thicket of tissue.
No, it doesn't. They had to somehow make only the astrocytes visible.
So how do you paint a single cell inside a living brain?
To do that, they engineered specialized glowing proteins. They used viral vectors, specifically adeno-associated viruses, or AAVs, to deliver them exclusively into the astrocytes.
Wait, they used a virus to carry glowing paint into the cell?
Basically, yeah. They used one specific tag, AAV25GOABC quantity tomato, which is a long name. But what it does is it fills the entire inside of the cell, the cytosol, with a brilliant glowing red.
Wow. Okay, so inside is red.
Right. Then, to be absolutely certain they were tracking the physical boundaries of the cell, they delivered a second tag. This one was AAV25GFA-ABC1DLEKEGFP.
And what did that one do?
That painted the outer cellular membrane a bright, glowing green.
Oh, that's wild. So with the red interior and the green boundary, they essentially created a neon sign out of every single astrocyte.
They really did. But these cells aren't just simple spheres. Like we said, they are star-shaped for a reason.
Right. They have those thousands of tiny, feathery branches extending in every direction.
Yeah, and measuring the volume of something that complex requires a lot more than a flat photograph. The researchers had to continuously map these glowing cells into full three-dimensional volumes.
Which sounds like a math nightmare.
It is. Picture an onion. You can't know its total volume by just looking at the surface. You have to understand every layer. So the scientists mathematically sliced the astrocytes into concentric 3D spheres.
Okay, concentric spheres, like measuring it from the inside out.
Exactly. They measured the volume right at the dense center of the cell, then stepped out 10 micrometers and then 20 micrometers, all the way to the very wisty outer edges at 30 micrometers from the center.
That is just an astonishing level of precision. But if I'm a skeptical scientist reviewing this paper, I'm asking how they know their microscope isn't just tricking them.
Oh, sure. You have to prove the tool works.
Right, like how do we know a fluctuation in that neon green boundary isn't just an optical illusion or like a focus issue with the lens?
Any rigorous experiment requires a positive control. You have to intentionally force the biological system to do the thing you were trying to measure, just to prove your instrument can actually see it.
So what do they do to force the cells to swell?
The researchers deliberately induced water intoxication in the mice by injecting distilled water into their systems.
Because distilled water lacks the natural salts and electrolytes of light.
Exactly. So through basic osmosis, the body tries to balance the concentration by shoving that excess pure water inside the cells,
Which makes them swell up like balloons.
the brain tissue swelled significantly. And when the researchers looked through their 2-photon microscope, they watched the astrocytes expand by 23.75%.
Almost a quarter of their size.
Yeah. It provided undeniable visual proof that their setup could capture real-time cellular swelling with incredible accuracy.
Wait, I have to push back here because this brings up a massive physical paradox.
Okay, what is it?
The skull is a rigid bone box. It does not give. It does not expand. So if you have millions of these star-shaped astrocyte cells simultaneously swelling up by almost a quarter of their size,
Yeah.
where is that physical volume coming from?
It's a great question.
Doesn't that completely crush the space around them?
That is the crucial mechanical reality of the brain. It is a strict zero-sum game inside the skull. When the astrocytes swell and demand more physical space, that extra volume has to be stolen from somewhere else.
So who gets robbed?
The interstitial space, those fluid-filled highways we mentioned earlier, that is what pays the price. The fluid is physically absorbed from the outside of the cell to the inside, crushing the highways down to narrow alleys.
Man, the physical trade-off there is massive. So let's pause and synthesize what we've uncovered in this first part.
Sounds good.
We've got these incredibly complex star-shaped support cells called astrocytes. We know scientists can use infrared lasers and viral glowing paint to watch them in 3 dimensions inside a living brain.
Right.
And we know that when these cells inflate like sponges, they physically crush the fluid pathways between them.
That sums up the mechanics perfectly. So I want to pose a question to you, the listener. Think about your own daily rhythm.
Yeah, picture your own day.
When you feel rested versus when you feel active. Based on what we just discussed, do you think these microscopic sponges in your brain inflate when you are deeply asleep or when you are wide awake?
It feels completely counterintuitive because most of us associate resting with expanding, taking a deep breath, letting your muscles relax, inflating.
It's a natural assumption.
But the data reveals the exact opposite. Your astrocytes are actually at their absolute largest, their most swollen, when you are fully awake and alert.
Yeah, the researchers proved this by continuously tracking the mice through natural sleep-wake cycles, monitoring their brainwaves with EEGs.
So what did the data show for the waking state?
During wakefulness, the astrocyte volume hits its absolute maximum peak. Every cell is swollen, and the fluid highways between them are squeezed tight.
But the transition into sleep isn't just an on-off switch, right? It happens in stages.
Exactly. When the mice drifted into non-rapid eye movement sleep and REM sleep, which is that deep, restorative, slow-wave phase, the astrocytes began to deflate.
By how much?
The total volume of the cells shrank by 8.14%.
Over 8% is a lot of physical space opening up inside a rigid skull.
It is, but that isn't even the floor.
Wait, really? It goes lower.
Oh, yeah. As the mice transitioned into rapid eye movement sleep, or REM sleep, that highly active phase where vivid dreaming occurs, the astrocyte volume absolutely plummeted. It dropped by 21.46% compared to the waking state.
A fifth of the cell's volume just vanishes during REM sleep.
It's just gone, absorbed back into the fluid highways.
That is a colossal architectural shift happening inside your head every single night as you cycle through different sleep stages.
And, the researchers didn't always need to drill down with lasers to know this was happening. They found a brilliant visible proxy staring right back at them.
Oh, right. The pupil.
Yeah, pupil diameter is actually a highly reliable indicator of cortical alertness. The autonomic nervous system dilates the pupil when you are highly alert or stimulated.
And how did that relate to the sponges?
The researchers mapped pupil size directly against their microscopic imaging and found a perfect positive correlation.
So bigger pupils literally equal bigger astrocytes.
It gives you a macro window into a micro world. You can essentially look into someone's eyes, observe the dilation of their pupils, and accurately predict just how swollen the millions of astrocytes are deep inside their cortex.
Here's where it gets really interesting, though, because measuring natural sleep is one thing, but they wanted to see how the system reacts to extreme artificial manipulation.
Right. They didn't just want to watch natural cycles.
So they introduced isoflurane anesthesia.
Anesthesia is a fascinating tool for neuroscientists because it forces the brain into a profound state of unconsciousness much faster than natural sleep ever could.
So what happened when the gas hit?
When they administered the isoflurane gas, the astrocytes didn't just slowly deflate, they crashed. The cells shrank by 17.9% in under 3 minutes.
3 minutes? That's not a gentle biological tapering. That is a sudden structural collapse of the cells.
It's violent, and then they cut the gas off.
Oh, to see how it recovers.
The rebound was even more violent. As the anesthesia cleared and the brain raced back toward wakefulness, the astrocytes rapidly reinflated, expanding by 20.55% in just 11 minutes.
Yeah. It paints a picture of a brain that is violently morphing its internal structure to match its level of consciousness.
It really does.
So if we pull together these threads on the sleep-wake cycle, let's do a quick recap.
Go for it.
Astrocytes are fully inflated when you are awake. They shrink by over 8% during deep NREM sleep, and they drastically deflate by over 21% during REM sleep.
Spot on.
We also know that you can use pupil size as a real-time monitor for this swelling, and that chemical interventions like anesthesia can force this inflation and deflation to happen in just a matter of minutes.
Which brings us to a profound biological mystery. Think about it. An astrocyte cannot see the outside world. It doesn't know what time it is.
Right.
So, to you listening, what is the messenger? What specific chemical signal floods the brain and tells millions of individual star-shaped cells to instantly inflate the second your alarm goes off in the morning?
Yeah, there has to be a physical trigger. A cell doesn't just spontaneously decide to swell by 20% on its own.
The researchers suspected a neurotransmitter called norepinephrine, often abbreviated as NE.
Norepinephrine. What's its usual job?
It's one of the brain's primary wake-promoting chemicals. It's manufactured in a tiny, highly specialized cluster of neurons deep in the brainstem called the locus coeruleus.
Okay, the locus coeruleus makes NE, but if they suspected it, they couldn't just assume it was there, right? They had to measure its presence in the interstitial fluid at the exact same moment the cells swelled.
Right, the timing had to match. So they employed a technique called fiber photometry.
Fiber photometry, how does that work?
By implanting a micro-optical fiber alongside a genetically encoded biosensor called GrabNE2MA.
GrabNE2MA.
Yeah. And with this, they could literally watch the chemical environment light up. This biosensor is engineered to glow whenever it physically binds to norepinephrine.
Oh, that's clever. It's like having a molecular ticker tape, constantly printing out the exact concentration of the chemical in real time.
Exactly. And the readouts mapped perfectly onto the shapeshifting.
Perfectly.
Perfectly. High norepinephrine during wakefulness meant swollen astrocytes. Lower levels during NREM sleep meant moderate shrinkage. And when norepinephrine almost completely vanished during REM sleep, the cells were fully deflated.
The two data sets moved in perfect, undeniable lockstep. The ambient concentration of norepinephrine dictated the physical size of the astrocyte.
That's what the data showed.
I pictured norepinephrine functioning almost like a specialized alarm clock. But instead of just waking you up with a noise, it physically inflates the room you are sleeping in, forcing you to stand up.
That's a great visual. But in science, seeing two things happen at the same time isn't enough. Correlation doesn't prove causation.
Right, standard scientific method.
Yeah.
They had to prove that the norepinephrine was the actual key turning the lock.
And to isolate the mechanism, you have to break it. You have to block the signal and observe if the shape-shifting stops.
So they threw some drugs at it.
They used a pharmacological blockade, injecting a cocktail of antagonists known as PPA. This cocktail blanketed the brain, blocking all the major receptor types that norepinephrine usually binds to, the alpha-1, alpha-2, and beta-adrenergic receptors.
And what happened?
When all those receptors were blocked, the mouse woke up, but the astrocyte stayed completely flat.
Wow. So the norepinephrine was there, floating around, but it couldn't turn the locks.
Exactly. Then they had to find out exactly which of those three locks was the right one.
So they tested them one by one.
First, they administered propranolol, which blocks the beta receptors.
Wow.
The astrocytes still expanded.
Okay, so not beta.
Then they tried atipamezole, blocking the alpha-2 receptors. Again, the astrocytes expanded normally.
That leaves just one lock remaining.
Yep. When they injected a drug called prizocin, which selectively blocks only the alpha-1 adrenergic receptor, the system completely stalled. The astrocyte expansion was entirely shut down.
Boom. Prizocin is the smoking gun.
It is. But Deng and his team didn't just stop at injecting drugs. They brought in some tools that sound completely like science fiction to double-check their work.
Optogenetics and chemogenetics.
Yes. Let's look at optogenetics first because the visual of this is incredible.
I'm going to break it down.
Optogenetics allows scientists to turn specific neurons on and off using light. They genetically altered only the norepinephrine-producing neurons in the locus coeruleus, the LCTH plus neurons, making them sensitive to light.
Okay.
While the mouse was in deep, slow-wave NREM sleep, the researchers aimed a tiny 470-nanometer blue laser directly at those neurons.
A blue laser inside the brain. And the instant that blue light hits the tissue, those specific neurons fire, right? Flooding the brain with norepinephrine.
Yes. The mouse snaps awake, and within seconds of the laser turning on, the astrocytes massively expand.
That is just wild.
And they reproduce the exact same biological response with chemogenetics using something called DREDDs.
DREDDs, designer receptors exclusively activated by designer drugs.
Exactly. Here, they engineered the neurons to react to a completely synthetic chemical called CNO. Injecting CNO forced the neurons to dump norepinephrine, triggering a massive 23.3% expansion of the astrocytes.
But the most important part of these high-tech laser and synthetic drug experiments is what happened when they pre-treated the mice with prizocin.
Right. Even when they blasted the locus coeruleus with the blue laser or flooded it with the designer drug, if that alpha-1 receptor was blocked by prizocin, the cells refused to swell. What's fascinating here is the absolute mechanical certainty this provides. The alpha-1 receptor isn't just loosely associated with the process. It is the direct, undeniable gateway.
It's the primary lever.
It is the primary lever that norepinephrine pulls to physically inflate the brain's architecture.
So to recap our detective work on this chemical trigger. Bullet 1. We know norepinephrine is the signal telling the astrocytes to swell.
Yep.
Bullet 2. We know the alpha-1 adrenergic receptor is the exact lock that norepinephrine must turn to make that happen.
Right.
Bullet 3. We know that if we jam that lock with a drug like prizocin, the brain simply cannot shapeshift, regardless of how intensely we stimulate it with blue lasers or synthetic drugs.
Which forces us to ask the ultimate question for our listener. We know how they swell, down to the molecular receptor. But why do they swell?
Why did evolution design this incredibly complex, energy-intensive system to physically warp our brain cells every single day?
It doesn't make sense unless there's a huge payoff.
Right. So what does this all mean? The body doesn't waste energy on this scale without a critical functional purpose. So where does this swelling actually happen the most?
To find the why, we have to revisit how they measured the cell in those 3D concentric spheres. Remember the onion layers?
At 10, 20, and 30 micrometers out.
Exactly. The researchers discovered that the core of the astrocyte barely changes. The massive volume shifts happen almost entirely in the distal processes.
The distal processes.
Okay.
Meaning the very tips of the star.
Right. Those feathery outer fringes 20 to 30 micrometers away from the center.
Why is that specific outer territory so important?
Because those outer fringes are intricately tangled up with the brain's synapses. Synapses are the microscopic junctions where neurons pass chemical and electrical signals to one another.
Okay.
When you wake up, norepinephrine floods your system, and those feathery astrocyte tips swell up, wrapping themselves tightly around the synapses.
Oh, I'm picturing an electrician wrapping a thick layer of electrical tape around a spliced wire.
It serves the exact same purpose, physical insulation. When a neuron fires a waking signal, often using a neurotransmitter called glutamate, that swollen astrocyte barrier prevents something called glutamate spillover.
Glutamate spillover, meaning it traps the chemical messenger inside that one specific synapse.
Yes, ensuring the signal doesn't leak out into the surrounding tissue.
That makes perfect evolutionary sense.
Yeah.
I mean, when we are awake, we need our cognition to be razor sharp. If you are tracking a moving object or trying to understand complex speech, you need high-fidelity, independent signals.
Exactly. If the glutamate leaks out and accidentally triggers the neighboring neurons, your brain would be flooded with static and crosstalk.
So the swollen astrocytes ensure that your waking thoughts remain crisp and independent.
That's the daytime shift. But let's look at the inverse. What happens at night when the norepinephrine drops and those astrocytes deflate?
Well, if the electrical tape shrinks and peels away from the wire, the signal is going to leak.
And during sleep, that leak is exactly what the brain wants. As the astrocyte shrinks, the interstitial space expands. Without that tight physical insulation, you get widespread transmitter spillover.
So instead of isolated independent firing, the neurons start talking to everyone at once.
They do. When one neuron fires, the chemical drifts across the expanded fluid highways and gently activates neighboring synapses. It creates synchronization.
Oh, wow.
This deliberate spillover is the mechanical driver behind the slow, rolling delta waves you see on an EEG during deep sleep. Millions of neurons binding together in a slow, rhythmic chorus instead of sharp, independent spikes.
The physical geometry of the brain literally dictates the type of brainwaves it can produce.
It completely governs it.
That is a staggering concept. But generating slow waves isn't the only benefit of those fluid highways opening up at night, right? There's a critical maintenance function here, too.
Absolutely. We have to talk about the glymphatic shift. This is essentially the brain's plumbing and waste disposal network. Throughout the day, normal neuronal firing generates a tremendous amount of metabolic waste.
Like exhaust from a car engine.
Right. And one of the primary byproducts is a protein called amyloid beta, which is notoriously associated with Alzheimer's disease.
Oh, man. So if your astrocytes are heavily swollen all day to keep your thoughts sharp, those interstitial highways are crushed down to narrow alleys.
They are completely blocked.
I'm imagining a crowded room where everyone has puffed out their chests and is standing shoulder to shoulder. The janitor is standing at the door with a broom, but he physically cannot get into the room to sweep the floors.
That's exactly what it's like. But the moment you fall into deep NREM sleep, norepinephrine drops and the astrocytes shrink.
Everyone in the crowded room exhales and sucks their stomachs in.
Suddenly, the pathways open up. Cerebrospinal fluid rushes through those widened interstitial highways, flushing the accumulated amyloid beta and metabolic toxic waste out of the brain. If we connect this to the bigger picture, this physical cellular breathing perfectly bridges the microscopic chemistry of sleep with the macro behavior of the human brain.
Right. Your ability to think clearly, to sleep deeply, and to wash away disease-causing proteins is fundamentally tied to the physical volume of your astrocytes.
Let's contextualize this for the listener with something everyone has experienced. Have you ever been abruptly woken up from a very deep sleep, maybe by a loud alarm, and you feel incredibly groggy and confused?
Oh, sleep inertia. It feels like your brain is wrapped in cotton. You can barely put a sentence together.
Based on this paper, that grogginess makes perfect mechanical sense.
Really? How so?
When the alarm sounds, your norepinephrine spikes. But it takes time for millions of shrunken astrocytes to physically inflate and wrap around your synapses.
Oh, until that electrical tape is securely back in place, your waking circuits are still experiencing transmitter spillover and crosstalk.
Exactly. Your brain is physically still trapped in its sleep state geometry.
It totally reframes the entire experience of waking up. Like, my sponges just haven't inflated yet.
Exactly.
Well, we've covered an immense amount of ground today. We've seen how infrared lasers reveal the breathing of star-shaped cells. We've learned that norepinephrine uses the alpha-1 receptor to inflate these cells during the day, insulating our synapses for sharp cognition.
And we've seen how their shrinkage at night opens the hallways for synchronized sleep waves and the vital flushing out of Alzheimer's-linked waste.
It really is a complete paradigm shift in neuroscience. And this raises an important question, one that the authors themselves hint at in their conclusion. If this daily shape-shifting is non-negotiable for a healthy mind, what happens when the mechanical machinery breaks down?
What happens if the alpha-1 receptor malfunctions, right?
Yeah, like if the lock rusts over or the key breaks off in the door.
Consider the clinical implications. If the astrocytes fail to properly deflate during sleep, the brain can't wash away the toxic amyloid beta.
Leading to neurodegeneration.
Right. But even more acutely, what if they fail to swell when you are awake?
If you lose that synaptic insulation.
The widespread crosstalk and electrical leakage could be the hidden physical root behind abnormal circuit activity. We could be looking at the structural foundation of psychiatric conditions like acute psychosis, sudden delirium, or even the synchronized electrical storms that cause epilepsy.
Wow. It suggests that treating neurological diseases in the future might not just be about adjusting the electrical firing of the neurons.
No, we might need to focus on repairing the sponges that insulate them. It's a completely new frontier for medicine.
To you, our listener, Thank you for coming on this deep dive journey with us. Tonight, if you happen to look over at your sleeping pet or your partner resting next to you, take a second to imagine what is actually happening beneath the surface.
Yeah, those rigid anatomical blueprints are gone.
Picture the microscopic tide of millions of star-shaped cells quietly receding, the fluid highways opening up, and the brain physically reshaping itself in the dark. Thanks for listening.